Welcome back, everyone. I'm Robin from This Blog's Neat, and if you clicked on this video, then you're probably a fan of funky Jamaican rums, or at least you're curious about where Jamaican rums get their funkiness. Now, as you know, I am a huge fan of funky Jamaican rums, and no, not all rums in Jamaica are funky, and it's not only rums in Jamaica that are funky. However, if you want funk, you go to Jamaica. Now, I have looked into the production practices that are used in Jamaica that produce these really super funky, high ester Jamaican rums. But I wanted to understand actually on a biological level, right? A biochemical level, how is this happening? How is this working? Why does it work? And why do these production processes work? So let's dive into all of that. We're gonna dive into actually how yeast produce esters, what esters are in the first place, and how all the environmental factors and everything that goes into a Jamaican rum, high ester Jamaican rum fermentation result in these high ester rums. So we're gonna dive into that today. And before we dive in, I do wanna give a huge shout out to my Patreons. Thank you guys so much for helping to support the channel. And you know that I've been reading tons and tons of papers to try and understand ester production, ester formation by yeast in the context of Jamaican rum, high ester rum fermentations. So if you, the viewer, would also like to join us on Patreon, I'd be so grateful. I've got a link in the description below where you can join us there. So yeah, let's get started. Jamaican rums are so funky because they are high ester rums, meaning they have a high concentration of esters. Esters are these fruity and floral flavor and aroma compounds that are produced by the condensation reaction of a carboxylic acid or a fatty acid and an alcohol, right? So this means you take a chemical compound that has a pretty acidic smell and taste, right? Think of acetic acid, which is like vinegar, and you combine that with an alcohol like ethanol, which is an alcoholic smell and taste. When you combine those via an esterification reaction, you get an ester that now tastes floral and fruity. So much better. And during fermentation, yeast are the ones who are responsible for esterification. This esterification reaction is catalyzed by enzymes within the yeast. Esters, by the way, are present in all fermented beverages, not just Jamaican rums. However, Jamaican rum producers have figured out how to just dial up that ester production a lot. So the production techniques used to produce high ester Jamaican rums are commonly long duration fermentations, spontaneous fermentations, and the use of both Dunder and Muck in the fermentation. I have also discussed the use of Dunder and Muck in Jamaican rum fermentations in detail in these videos right here. Go ahead and check those out. You'll get more details about Dunder and Muck. Now keep in mind, not every distillery in Jamaica does it this way, and that's why you have varying amounts of funkiness, right? Varying amounts of high ester quantities. And a lot of these distilleries will have various marks, right? So they'll do some batches that are super funky and others that are really clean. That way they can blend those together and create a much more balanced flavor profile. So some distilleries may use just long duration fermentations. Some distilleries might use spontaneous fermentation. Others might use Dunder and Muck, and some of them, like Hamden Estates, will use all of those techniques to create super funky Jamaican rums, super high ester Jamaican rums. Now this all happens because yeast are the ones who are producing the esters. So let's talk about how yeast produce esters in the first place. First of all, they are creating two different types of esters. They're producing acetate esters and ethyl esters. Acetate esters are derived from a reaction between acetic acid and an alcohol, and ethyl esters are by combining ethanol with a fatty acid or carboxylic acid. So there you have it, these two different types of esters that are produced, and they are produced via different mechanisms and different enzymes within the cell. Here are some examples of acetate esters, and here are some examples of ethyl esters. Now, the reason that I bring up these two different types of esters is because acetate esters diffuse completely and very quickly through a cell. This means as soon as the yeast produce them, they get expelled into the medium. Whereas with ethyl esters, that depends on the chain length, right? How big, how bulky is the molecule. For instance, ethyl hexanoate will diffuse completely, 100% diffusion, whereas ethyl decanoate's diffusion rate is anywhere between like 8 and 17%. This is, of course, in Saccharomyces cerevisiae, which is the yeast that is most commonly found for brewing, distilling, wine making, bread making even, right? Saccharomyces cerevisiae, this is our most common fermentation yeast, the most common yeast that's also studied. Now, acetate esters and the mechanisms and genes that are responsible for producing acetate esters have been studied a lot more, or at least have been a lot easier to study, whereas ethyl esters, it kind of depends on what studies you look at. But I will give you a direct quote from a paper published in 2023, so that is this year. The mechanism, including genetics and regulation of fatty acid ethyl ester formation, are still less clear. The ATF1 and ATF2 genes are what are most involved in acetate ester production, and this was figured out by deleting and then overexpressing the genes. So when both of those genes, the ATF1 and ATF2 genes, were completely deleted out of the cell, the amount of esters that were produced, acetate esters that were produced, dropped significantly. Interestingly, it was not dropped to zero. There was still some acetate ester production, so keep this in mind. Now, when both of those genes were overexpressed, there was a lot more acetate ester production. Ethyl esters, on the other hand, it's not so clear. However, the two genes that are proposed to be the most responsible for ethyl ester production are the EHT1 and EEB1 genes. Interestingly, deletion of both of those genes decreases the amount of ethyl esters produced, does not eliminate the amount of ethyl esters produced. However, overexpressing those genes does not change the amount of ethyl esters that are produced. I think that's really interesting. So this kind of means that those genes are not the limiting factor in the amount of ethyl esters that are produced. So after reading a bunch of these studies, trying to figure out what genes are the ones that are responsible for ester production, both acetate and ethyl ester production, you can see that deleting the genes, for the most part, didn't eliminate ester production outright, right? So ester production is deeply ingrained somehow in the basic cell function, right? Again, these studies were done on Saccharomyces cerevisiae, various strains of Saccharomyces cerevisiae, but still brewer's yeast, right? So I wanted to know, why are yeast producing esters? Why is this so important for their biological function? And luckily there is a paper that has outlined the 4 different hypotheses that are out there as to why yeast produce esters. Of course, you can't just like ask a yeast cell, why are you doing this? But there are some hypotheses out there. The first hypothesis is that ester formation frees up CoA. This is coenzyme A, which is a key factor in a number of different functions within the cell. The second hypothesis is that esters can act as unsaturated fatty acid analogs, which really just means that esters can help the cell with fluidity of the cell membrane. The third hypothesis is that esters can act as a detoxifying mechanism in the presence of medium-chain fatty acids, which are able to diffuse into the cell. So say there is an environment where the medium is filled with a lot of medium-chain fatty acids that are able to diffuse into the cell. Medium-chain fatty acids are toxic to yeast cells. However, medium-chain fatty acid ethyl esters are not, or at least they're less toxic to the yeast cell, and they diffuse more readily across the cell membrane. They're more lipid soluble. So that means the yeast, if it's taking in medium-chain fatty acids, or if there are any present within the cell, it is able to convert those to ethyl esters and then expel them. Now, the fourth hypothesis is that ester formation helps with yeast dispersion into nature. So bugs, namely like flies, are really attracted to esters. And because yeast can't just like pick up and move and fly away somewhere else, they need a little help. So they can hitch a ride on any flies that are attracted to the fermenting medium. That way they can get carried to other places and their species can continue to thrive on. So in summary, ester formation just like helps out the yeast in a number of different functions. And it is probably the purpose of why yeast do this is probably a combination of all four of these hypotheses and not just one, right? Okay, so now that we know how esters are produced by yeast, why they're produced by yeast, let's see how ester formation is affected by environmental factors. So first and foremost, the yeast species and yeast strain will have a huge impact on what esters and how many esters are produced, right? This is in the genetic makeup of the yeast. So all these studies that are done looking at Saccharomyces cerevisiae show that even from a wine strain versus a brewing strain, there are going to be differences in the amount of esters and the types of esters that are produced. And we kind of know this as brewers and distillers, right? We know that there are varying types of Saccharomyces cerevisiae, varying strains that will give us different ester profiles. But that also means that within species, right, Saccharomyces versus Pichia species, they activate these genes differently, these ester producing genes differently. For example, in Saccharomyces, the ATF1 gene, which is most responsible for acetate ester production, is suppressed in the presence of oxygen. So in aerobic environments, like say the beginning of a fermentation, Saccharomyces cerevisiae or Saccharomyces species are not producing a lot of esters, acetate esters. Whereas in Pichia species, the ATF1 gene is expressed in aerobic environments. So even in the same environment, Saccharomyces species versus Pichia species, one will not produce esters or very little esters, and the other one will be producing a lot more acetate esters. Environmental conditions also have a huge impact. Temperature, for instance, will impact the amount of esters that are produced. So generally speaking, an increase in temperature will cause Saccharomyces cerevisiae to produce an increase in esters just across the board, ethyl esters and acetate esters, with the exception of ethyl hexanoate. That's the only one that saw a little decrease in this one study that I was looking at. The carbon to nitrogen ratio will also impact the amount of esters that are produced. So the amount of nutrients that are in the medium will have an impact on these esters. For ethyl esters, it's pretty variable. However, with acetate esters, an increase in nutrients, you see an increase in acetate esters. The amount of unsaturated fatty acids within the medium will also impact ester production. So this is like the difference between doing a brew using a clear wort versus a cloudy wort, right? The cloudy wort has more unsaturated fatty acids, and a higher concentration of unsaturated fatty acids causes a decrease in ester production across the board. Keep in mind, these studies that I just spoke about, all done on Saccharomyces cerevisiae. Now, another thing that has a huge impact on ester production is the other microbes that are present. So if it's not just Saccharomyces cerevisiae in your brew, you're gonna see a wide variety of implications. One study that I looked at co-pitched Saccharomyces cerevisiae with Hanseníaspora uverum in a wine fermentation and observed an increase in medium chain fatty acid ethyl esters. But that's just pitching two different types of yeast. And depending on the other yeast, that is not exactly what is observed. So it's really dependent on what other yeast is there. Now, I read another interesting study that co-pitched Saccharomyces cerevisiae with a bacteria, Lactoplantobacillus plantarum, which I am going to call L. plantarum from now on. So in the study, L. plantarum had a very antagonistic relationship with S. cerevisiae, where it inhibited its growth. It also downregulated a whole bunch of basic functions, cell functions, and then upregulated a bunch of stress responses. In the study, they also observed that the L. plantarum surrounded the S. cerevisiae, like it was physically surrounding the yeast cell. They also observed that in the co-pitched fermentation, L. plantarum lambdca quorum sensing system was activated. Now, quorum sensing is how bacteria communicate with one another. They send these signaling molecules called autoinducers back and forth. They can communicate with other bacterial species. They can communicate with themselves. It's really cool, and I want to dive more into quorum sensing after reading about this. Maybe it's just me. Maybe I'm just like, how do yeast and bacteria know that one another is around, right? They don't have eyeballs, they don't have brains, they can't they don't have the sense of touch. They're just these single-celled organisms that are able to sense one another through chemical signaling. And yeah, I find that really fascinating. And this chemical signaling will dictate what genes to turn on, what genes to underexpress, overexpress, etc. But essentially, they hypothesized that they upregulated this quorum sensing system as a way to increase adhesion of the bacteria to the yeast cell. Anyways, Robin, why do you bring all of this up? You're probably asking. In Jamaican rum fermentations, if they're adding in Dunder and Muck and doing spontaneous fermentations, the amount of microbes that are in that fermentation is vast, right? And this study that I'm referencing, they're just looking at two microbes, the interaction between two microbes. So imagine what craziness is going on inside a fermentation for a high ester Jamaican rum. It's crazy. So if we zoom back out and look at this high ester rum fermentation, we'll say it's using spontaneous fermentation, long duration fermentations, Dunder, Muck, and it's in this warm, humid environment. You have a number of ways where the yeast that are producing your esters are just expressing these stress responses. First of all, there's tons of other microbes that are in there, which is going to cause yeast to behave differently. So I've referenced this paper before in another video about Dunder and Muck, or about Jamaican rum fermentations, but this is the microbial flora of rum fermentation media. This is referencing rums that are produced in Haiti using slops, which I believe is more like a Dunder type medium. They listed 18 different yeast species that were found within these rums, as well as like 30 different bacteria species. So So that's a lot going on there. The use of Dunder and Muck is adding in a bunch of medium chain fatty acids, which, as we've discussed, is toxic when it diffuses into a yeast cell. And so they want to kind of get rid of that and can convert that into medium chain fatty acid ethyl esters and then diffuse that back into the medium. High temperatures, which is going to increase your ester production. There's also long duration fermentations, which can have a number of effects, namely like stressing out the yeast, right? As the fermentable sugars are converted into ethanol, the yeast is starting to die off and they need to propagate their species. So they're going to upregulate some of those ester producing genes in the hopes that flies will carry them away. Longer duration fermentations also allow for just more of those medium chain fatty acids to diffuse into the cells and get converted more into ethyl esters. So there you go, guys. That is how yeast produce esters. That's why yeast might be producing esters. That's how Jamaican rum fermentations, these spontaneous, very microbially diverse fermentations, are causing this high ester production. I hope I did a good job conveying how cool I think this is. And it's super complex, but I think it's really interesting. And the reason that I share this with you is that I hope that you now have generated a little bit more appreciation for all of the chemical reactions that are going on in the fermentation, all of the biochemical reactions that are going on that result in not only esters that are in our high-ester Jamaican rums or high-ester rums, but are also in all fermented beverages that we enjoy. So if you have any questions at all, please let me know in the comments below. If I explained something and it was unclear, please let me know. If you want references to any of the papers that I've read, which I only held up 2, but I have a binder of papers next to me and another stack right here that I've gone through all just to understand how, why, what's going on here. Yeah, let me know and I can give you references to any of the papers that I have read or that I found really interesting. Now, when yeast produce esters, they are, what are you doing on the stairs? With Saccharomyces cerevisiae, and noticed, yeah, do you see a bird? Yeah, you do. You do. Lactoplantibacillus plantarum, and lactoplantibacillus plantarum, and lactoplantibacillus. Oh, I promise I can say this. They also will use Dunder, which is the leftover stillage, so the liquid that's left over in the still after distillation that has a lot of acidic compounds in it. They'll toss that back in. Keep in mind, this is not unique to Jamaican rums, right? This is not unique to rums. It's done in bourbon all the time. They call that sour mash or backset, right? And they're adding that back into their fermentations. Muck is also something that's used in Jamaican rum production. And this is a lot more microbially diverse, if you will. This sits out for a lot longer. It grows a lot of bacteria, other yeast. It's filled with tons of these medium chain fatty acids that have quite putrid smells to them, but later on can turn into some really beautiful esters. Anyways, Muck has the lees tossed in from fermentation, the sediment that's left in the bottom of the stills after distillation. It has cane trash tossed in. It's rumored to have, fruits tossed in and everything like that. And I have told a number of stories about what has gone into Muck that I'm not sure holds any actual truth to them, but Muck is a lot more, as I said, microbially diverse. And Dunder, on the other hand, depending on how quickly it is used after distillation, can have some microbial diversity, but once it's distilled and everything, once it comes out of the still, if it's used fresh, it should be pretty sterile. However, there's a chance that Dunder is sitting around for a while and can then pick up some of the ambient microbes